Hello and once again welcome to the program of Bachelor of Science second year, subject Botany, semester 4, course code is BOC 104 and the course title Plant Physiology. Title of the Unit 4 Enzymes, and the name of the module is Enzyme Inhibition Part 2. In this module, I'll be explaining irreversible inhibition and allosteric inhibition. In the previous module, you have already studied about the reversible inhibition. At the end of this module, student defines irreversible inhibition, explains irreversible inhibition, defines allosteric inhibition, and explains allosteric inhibition. In the previous module, you have studied about the enzyme inhibition. What is enzyme inhibition? It is a process in which the inhibitor binds with an enzyme. As a result, there is decrease or suppressing of the enzyme activity. You have also studied the types of the enzyme inhibitions like reversible inhibition, irreversible, and the allosteric inhibition. Out of these three inhibitions, you have studied in detail in the previous module, reversible inhibition. And so, in this module, I'm going to concentrate on the irreversible inhibition and allosteric inhibition. What is irreversible inhibition? In this type of inhibition, the inhibitors covalently bind with the enzymes and inactivate them, which is irreversible. Now, in this inhibition, the important thing is we call it as irreversible because whenever the inhibitor binds with an enzyme, it binds with a strong bond. And when it binds, it modifies the amino acid residue which is needed for the enzyme activity. And so, it becomes a reversible inhibition. These inhibitors which bring about the irreversible inhibition are generally the toxic substances. Now, in the slide, you can see there is an enzyme molecule and the inhibitor has come and it has bind to the active site of the enzyme molecule with the help of a bond. Now, because of the binding of the inhibitor to the enzyme inhibitor complex is formed. The inhibitor has changed the conformation of the enzyme and as such, the substrate is not available, or rather the enzyme is not available to the substrate to get converted into the product. The inhibitor has completely blocked the active site. The substrate cannot bind to the active site. If the substrate binds, it binds to the enzyme inhibitor complex. And in this way, thus you will find that the catalytic activity cannot take place. The enzyme action is inhibited by the inhibitor and the substrate cannot get converted into the product. Now, this is a very good example of the irreversible inhibition where iodoacetate is an irreversible inhibitor of the enzymes like pepin and glyceraldehyde 3-phosphate dehydrogenase. Now, coming to the allosteric inhibition. In this type of inhibition, there is an allosteric inhibitor which will inhibit the enzyme activity by binding it to the allosteric site of the enzyme. So, in this type of inhibition, there is an enzyme molecule which will have the active site, but apart from the active site, it will have another site which is called as an allosteric site. And to this allosteric site, the inhibitor called as allosteric inhibitor will come and bind. And thus, it will either decrease or it will suppress the activity of the enzyme. Now, in this slide, you can see there is a glucose molecule. Glucose, with the help of the ATP, it gets converted into glucose 6-phosphate and ADP molecule is formed. The conversion of glucose into glucose 6-phosphate is with the help of the enzyme hexokinase. Now, the product that is glucose 6-phosphate, it acts as an inhibitor. And when it acts as an inhibitor, it will inhibit, or rather it will go and will bind to the hexokinase enzyme and will not allow another glucose molecule to bind to the hexokinase so as to form the product that is glucose 6-phosphate. So over here what you can see is the product itself is acting as an inhibitor to the enzyme, and thus the substrate, that is glucose, is not converted into the product. The allosteric inhibition is generally seen into the multi-enzyme pathways, and this is an example of the multi-enzyme pathway where you can see A is the initial substrate. It gets converted into B, which is an intermediate compound, with the help of the enzyme 1. Again, B gets converted into C with the help of the enzyme 2. C gets converted into another intermediate compound with the help of the enzyme 3. And finally, the D, which is an intermediate compound, with the help of enzyme 4, it gets converted or it forms the final product. So, in this pathway, there are 4 enzymes involved which will form a chain of reactions. Now, if the final product, the amount of the final product in the cell exceeds, then this product acts as an inhibitor. It will go and bind to the enzyme 1, which is a regulatory enzyme, and it will inhibit the activity of that enzyme. As a result, the substrate will not bind to the enzyme 1, and further intermediate compounds will not be formed. As a result, the final product will be kept in check in the cell. That is, the amount of the final product will be kept in check inside the cell. So, this is also called as feedback inhibition. So, this is the allosteric inhibition where finally the product acts as an inhibitor and inhibits the activity of enzyme. Now, what is the importance or the significance of enzyme inhibition? Number 1, it helps us to study the mechanism of catalytic activity of enzyme. Secondly, it helps us to understand the regulation of enzyme activity. And the most important thing is these inhibitors, they are used or they have the therapeutic applications, means these inhibitors can be used to treat or to cure number of diseases. These are the references for this E module. Thank you.